Okay, so students, till now we have covered all the steps of recombinant DNA technology. Now we are going to see how we are going to select, okay, the selection of transformants. For this, we have to know the characteristics of vectors. Okay, so whenever you are going to multiply your genes with the help of vectors, you need to check some special some special characteristics, okay, that your vector should have. So we'll see the vector characteristics. You should know, okay, all these characteristics, vector characteristics. So the first characteristic, it should have origin, okay, origin of replication. Now origin of replication is the site, okay, where the DNA polymerase is going to recognize, okay. Once this DNA polymerase is going to recognize the origin of replication, then only your plasmids will get amplified. So the first necessary thing is your vector should have origin of replication. The second thing, it should have selectable marker. What is the selectable marker? See students, there are some, the plasmids are not required for the survival of the bacteria, but yes, it gives the special characteristics to the bacteria. So it helps this bacteria to survive in adverse conditions. So there are some genes, okay, the plasmids, they do contain some genes, okay, the genes, they are called as resistance genes. So they contain some resistance genes, the resistance genes against antibiotics. They may have the genes, resistance genes against tetracycline, okay, antibiotic. They may have the genes resistant for the antibiotic ampicillin, okay. So here I am writing, they could have tetracycline resistance gene, okay. They may have ampicillin resistance genes. So they should have the selectable markers. The next third thing it requires is, this is called as multiple cloning sites. What is this multiple cloning site? This is also called as polylinker region. So if suppose this is your plasmid, okay, the plasmid should have the restriction multiple cloning site. Means it should have the sites for restriction enzymes, okay. So that restriction enzyme will recognize a particular, it should not have many recognition sequences for many restriction enzymes. It should have a particular, okay, for example, it should have a restriction enzyme site, recognition site for EcoR1, okay, for HIND, Haemophilus influenza HIND3, okay. So it should have the restriction sites, okay, the recognition restriction sites for particular restriction enzymes. So these three criteria, if suppose your vector is going to fulfill these three criteria, origin of replication, then selectable marker and multiple cloning site, then your vector is perfect, okay, for transformation. This here we will see how we are going to check, okay, we are going to select the transformants. Suppose till now what we have done is we have incorporated our plasmids into the cell, okay. Now we are going to check which cells are transformants, which cells are non-transformants. So we are growing here the cells on a media, let's say media containing antibiotic ampicillin. So what will happen, the cells will grow here, okay, the cells that contain the plasmids, okay, that gives the resistance. Some plasmids will be transformed, some plasmids will not be transformed. So let's suppose these two cells are transformed, so transformants. So what will happen? Now they have the plasmids have the genes for ampicillin resistance. Okay, so what will happen? There's a plasmid genes, they will help these host organisms to survive against this antibiotic. Okay, so what will happen? These cells are going to survive, but as you see, these cells, they do not have the plasmids in them, so the cells are susceptible to the antibiotics ampicillin, so these cells are going to die. So the cells, okay, that are transformants, they are going to survive, and the cells, they are non-transformants, they are going to die. Okay, so this is how we are going to select our transformants. So this is the end of our session. We are going to write down all the steps, okay, that are involved in recombinant DNA technology. The first step is isolation of DNA from source organism. Okay, the second is, what we did is fragmenting this DNA, okay, with restriction enzyme. Then the third is inserting gene of interest in vector. Okay, and the fourth one is transforming host cells with vector. Okay, this becomes your recombinant DNA. This is called as rDNA or recombinant DNA. Okay, and the last one is, now you have transformed, selection of transformants. And the last one is multiplication of host cells, where you are going to multiply your host cells into bioreactors. And the last is expression of gene.